Okay, can everyone hear me out there? Perfect.
And hear me?
Okay, so here we go. Malaria 101. So first of all, I want to thank Bill for the introduction. I want to thank Rotary for the honor to present, to allow us to present today. And Linnie and I are going to go back and forth today. So we're going to hopefully teach you a little bit about what you need to have a very successful day learning about malaria. Now, I want to begin with a land acknowledgement. So as a local Albertan, I would like to begin by acknowledging the rich cultural history of the Indigenous communities here in Alberta. In Calgary in southern Alberta, we pay tribute to the traditional territories of the Treaty 7 people and the Métis Nation of Alberta. And I personally would also like to express my gratitude to the First Nations of Treaty 6 territory and the First Nation of the Métis Nation of Edmonton. I'm grateful for the opportunity and the privilege to live, to work, and to learn on their ancestral lands.
And if I could add.
Oh, absolutely.
Coming from New York, which is the land of the Lenape peoples, I am a proud person to be able to be here and be on the lands of others, and I appreciate the acknowledgement.
Thank you, Linnie. Okay, so Malaria 101, this is what we have lined up for you this morning. We're going to start off sharing a bit about the colorful history of malaria. This is a disease that has been with us for a very, very long time. Then we're going to talk about where are we now? What is the epidemiology of malaria? Who gets malaria? Where do they get it? Then we'll talk a little bit about the biology. That's really important here to understand what causes malaria. What does it do when it causes disease? And then we'll look at opportunities for interventions, but also what are some of the threats that are emerging to those interventions. And then we're going to wrap up by considering those broader challenges to malaria control. And we will invite you as Rotarians to join the fight against malaria. And with that, I'm going to pass it over to Linnie to start us off with the history.
Okay, so the history of malaria. How old is it exactly? Well, the New York Times in 2010 reported that it was considered to be the most likely killer of that famous King Tut in Egypt. And if you think about it as we go along, he lived on the Nile, a place where there, well, let's just say some swampy lands. We'll get to that. And indeed, swamps and wet areas were associated with malaria. The Greeks and Romans were aware of the disease, and they would go to higher ground at times when there was more water. But we go back again to the ancients. Chinese medical writings described the several symptoms of malaria. And Hippocrates, who some call the father of medicine for his astute knowledge of signs and symptoms of disease and his use of examination, had noted that these fevers could be associated with splenomegaly, an enlarged spleen. So right here in Canada, also there were issues with this association of these deaths with swamp fever in the Rideau Canal. And as it said here, no one could escape from malaria. It didn't matter whether you were rich or poor. If you were in these areas, you might indeed die. So Alphonse Lavarance, a French person working in Algeria with the military, actually began to look at the blood of people that had these diseases. And these are his hand-written drawings of the parasite. And he said he believed that these organisms, which he could see in the blood, were the cause of these fevers. It took Ronald Ross to make the discovery that connected the mosquito itself to the transmission of the disease, so that the parasites that had been seen previously were transmitted from the mosquito to humans. And in fact, he made this discovery working with avian malaria, or with birds. And so now we have the connection that the disease is transmitted from mosquitoes to the human host. What more can we say about the disease? And how can we interrupt? Now, with this knowledge, just saying we don't need to get rid of swamps, maybe we need to do something about the mosquito vector. Well, here a little bit to the south, where I hail from, malaria in the United States goes back to the slave trade when they believe it was introduced. And in fact, what Ronald Ross had discovered was it was the Anopheles mosquito which was involved with the transmission. So if you introduce the parasites into an area with those specific mosquitoes, you can get disease. And if you get rid of them, you can eradicate the disease in a particular area. And so this shows the distribution of malaria in 1882, and then you can see that in the 1930s it was really mostly found in the southern United States. And indeed the CDC was founded, United States CDC, on eradicating malaria in these areas where they would have it not only because of what had happened with the slave trade, but with people going back and forth to train for war, they would bring malaria to these areas, and it was ultimately eradicated. This led to moves, of course, to have a worldwide eradication of malaria, as shown here in India. And perhaps a little bit of hubris, Paul Russell in 1955 said, well, keeping in mind the realities, one can nevertheless be confident that malaria is well on its way towards oblivion. Already as a malariologist, I feel premonitory twinges of loneliness, and in my own organization, I am now a sort of last survivor. I think our presence here shows that he was wrong, and I'm going to. But he had reason to hope. As you can see here in this map from 1900 to 2002, areas that were malaria-free were increasing, and it looked like we really were on our way, so not to be too hard on him. But I now turn it back over to Stephanie to talk about the current status, which is indeed why we are all here.
Thank you, Linnie. So what have we done lately is the question. So let's look at the last 25 years and look at some of the changes, some of the gains, but also some of the challenges that we've seen in malaria control. So what I'm going to show you here are a few maps, and these are maps that show the incidence of malaria. So that means the number of new cases or new infections of malaria per 1,000 population. And if we look at the year 2000, we can see that those malaria control efforts after World War II were successful in more developed parts of the world, but clearly failed in the tropics. We can see that there's a high incidence, so the brighter the color, the more red, that means there's more infections of malaria. So clearly sub-Saharan Africa is dominating, but we also find that malaria is prevalent in the Americas, in South Asia, particularly in India, Southeast Asia, and also some of the Pacific Islands. Now this is in 2000, and 2000 is a very important year because it marks the initiation of the United Nations Millennium Development Goals. And that was a concerted global effort to address 8 key priorities. And one of those related to infectious diseases, and specifically malaria. And the goal was to halt and reverse the incidence of malaria by 2015. So let's look how we did with that. So here's 2010, so we're partway through the MDG era. We can see some gains in sub-Saharan Africa, but we also see that aside from the Americas, the map has not changed significantly. Okay, now 2019. Again, we do have some gains. Now India has reduced its incidence. The Americas have made very good progress. Some changes, minor changes in Africa, but we're also seeing some resurgence of malaria in certain countries. And if you look at Venezuela, which I don't have a pointer, but it's on the top of South America there, we're seeing that this was a country that was on the cusp of eliminating malaria. And now, because of the political instability and the breakdown of the health system, there's a massive resurgence of malaria, and it's also having spillover effects in neighboring countries like Colombia. Now 2022, this is post-COVID, and we're still seeing a lot of red on this map. And in fact, the 2 countries, Nigeria and the Democratic Republic of Congo, still have the greatest burden of disease, just like they did back in 2000. So we did not meet the Millennium Development Goal. And in 2016, the World Health Assembly launched a new program called the Global Technical Strategy for Malaria, which had a much more aggressive goal. This goal was to reduce the incidence and mortality from malaria by 90% by 2030. Let's look at the numbers. So in blue, the solid blue line, now here we're focused on sub-Saharan Africa. We're still looking at incidence. The blue shows some of those gains that were made during the period of the Millennium Development Goals. We see that number of cases are declining. But if we look around 2015, 2016, that changes. And we now see that that curve is plateauing. And if we project out to 2030 and continue that blue line, we see that that projected incidence rate is almost 10 times higher than that target, which is shown in green, of that global technical strategy. We're also very concerned about changes in the trends with malaria mortality. So here, we're looking at different regions of the world. Now we're not looking at cases, we're looking at deaths. And these deaths are in the hundreds of thousands. So if we look by region, we see that Africa has the highest mortality rate from malaria, followed by Asia. If you focus in on the year 2000, we can see that during that period, 2000 to 2015, there were some very important gains made in reducing malaria mortality, But then around 2017 or 2018, now this is before COVID, we're starting to see a reversal. And the number of deaths is actually increasing. It continued to increase during the pandemic, and now it's plateauing. We want that curve to come down. So how do we get back on track for malaria control and elimination? So here's where the biology comes in. We need to understand what causes malaria and identify opportunities to intervene to ultimately eradicate this disease. So Linnie introduced you to the parasite that causes this disease. It's called Plasmodium. And this is a parasite that's not unique to humans. Many, many different types of animals actually get malaria. And just recently, there was an outbreak of malaria in swallows in central London. So this is not unique to us. But there are clearly 4 species that are restricted to infection in humans. And Linnie will talk about those in more detail. They're called Plasmodium falciparum, vivax, ovale, and malariae. But there's also some zoonotic species. These are species whose native host is not human. It's usually a non-human primate, like a monkey. But because of changes in land use and deforestation, we're now finding that, for example, forest workers are coming into contact with the monkey population. Those mosquitoes are allowing that transmission of what's a monkey malaria into a human population. And this is particularly alarming in Borneo, where we're seeing a very aggressive form of monkey malaria that is infecting humans. Now, I want to emphasize that a parasite is a very complex organism. This is not a virus that expresses a handful of genes that we can manipulate and make a vaccine very quickly. This is a very complex creature. It expresses over 5,000 genes. That's a lot of genes, and these genes are distributed across 14 chromosomes. That's a lot of chromosomes for an organism that you can't even see unless you're looking under the microscope. And the parasite has evolved all of this genetic complexity because it needs to infect 2 very different creatures, humans and mosquitoes. And to do that, it uses its genetics to be able to live in different types of cells, to infect different cells, and to be able to thrive as a species. So now I'm going to pass it back over to Linnie, who's going to tell you what happens when malaria infects us humans.
So, a deadly disease in humans, how does it do that, and where exactly does it go? Because ultimately what we want to do is not only look at how can we break the chain, if you will, between mosquitoes and humans in the environment, but what might we be able to do within the human host itself? So the malaria life cycle. The Anopheles mosquitoes, as mentioned, injects the parasite into the blood. These sporozoites then go to the liver, where they replicate many, many times. Now, the person doesn't know they're infected. They're completely asymptomatic at that point. The parasite then enters the red blood cells, remember where they were first noted. And then in those rounds of replication, that's when the symptoms begin, and that's where the deadly effects occur. Some of those parasites become the gametocyte stages, which are the sexual stages. Those then are taken up by the bite of another mosquito, and then they develop within the mosquito, and then they can be passed on to others. So again, in the humans, it's those red blood cell stages that are the problem. And when the parasites break out of one red blood cell and go to another, that's when you get those cyclical fevers that were noted by the ancients. So as seen here, the different types of malaria in humans can cause different periodicities of fever depending upon when they break out. So they may be hectic fevers, they may occur every couple of days or every 3 days. When I was an intern at Harlem Hospital, one of my colleagues happened to call in sick every time he was on call. We were on call every 3rd night. Now, we thought he was a great malingerer. Yeah, right, you're sick again. But one of the great grandfathers of medicine at Harlem Hospital, Dr. Marquez, says, "My boy, didn't you go home for a visit recently?" And sure enough, he had malaria. We were just glad he was back on call. But at any rate, those are the kinds of things that might give you a clue that your patient has malaria and give us some clue as to what type they might have. The one that we're most concerned about, though, is Plasmodium falciparum. This is a very aggressive, very deadly disease. Your patient can die within 24 hours. Hence, the call that any patient who's been in an endemic area with fevers is presumed to have malaria until proven otherwise, which means you really need to look at the blood, see if you see those parasites, or use other tests which can indicate that they're there, which Stephanie will talk about later. With a patient with cerebral malaria, they have certain signs and symptoms that we can look at, and many in this audience are all too familiar with: coma, seizures, bleeding. They may just feel terrible, awful. Laboratory and radiographic findings: acidosis, anemia. Their blood sugar may be low. Of course, they may have a very high parasitemia if you look. They may become jaundiced or yellow. They may have problems with their kidneys, pulmonary edema, and ultimately, multi-organ shutdown. So these beautiful little children could be harboring parasites. They may look fine, and then suddenly get very, very ill. Again, as many in this audience know. And the predominance of deaths occurs actually in young children, 5 and under, mostly in sub-Saharan Africa, and also affects pregnant women worldwide. It not only affects their health, but that of their babies, causing a lot of morbidity and mortality. So how might we treat malaria? And this isn't going to be a treatise on that, but just to give you an idea that there are drugs which can affect each one of the stages shown here. So those stages in the liver, which you don't realize when you're the patient, the hypnozoites, particularly in some of the different species that cause the malaria, P. vivax or P. ovale, they may stay there for many, many months. Primaquine and tafenoquine are drugs that we might use for them. For the asexual blood stage, where we have the symptoms of disease, and I'm using here treatments that are used mostly in endemic countries, artemisinin-based combination therapy, or ACTs, are first line. Chloroquine, if you're in areas only where there's sensitive parasites, and those areas are now very, very few. And in severe disease, artemisinin is the drug of choice. Now, low-dose primaquine, which is ACT, can cause a reduction in gametocytes, and again, if you can reduce gametocytes, you can reduce transmission. So, what are other ways that we might interfere with the spread of malaria and stop disease? And so I now turn this back over to Stephanie, who will talk about that pull and push of what we're trying to do.
Thank you, Linnie. So here we have our trifecta. We have the human host, we have the mosquito, we have the parasite. And remember, these mosquitoes and the parasite are vying for survival. But we also have opportunities to intervene in ways that we can try to break that life cycle. So what we're going to do here is kind of tease apart that life cycle and think about what are some of the ways that we can intervene, but also what are some of those challenges that we need to be really vigilant about. So let's start with the liver stage. Now, the liver stage presents a really tremendous opportunity to break the life cycle of malaria. At the top, when those sporozoites are coming in, there's very few of them. The mosquito injects only a few parasites. So it's a bottleneck. And that means that if we can block the malaria infection at that stage and prevent those liver cells from getting infected and going through this massive stage of replication, we can then prevent all of the onward effects of malaria. We can prevent the infection in the blood, we can prevent that uptake in the mosquitoes and that onward transmission. And our number one tool to try to block the liver stage is through vaccines. And we're very excited that in the last couple of years, we have 2 vaccines that have been approved by the WHO. They're called RTSS and R21, and I know you'll hear more about R21 later today. There's also another approach for this vaccine, which is based on growing these parasites in mosquitoes in the lab and then basically disabling them so you can expose the immune system to all of the different components of the parasite. But the vaccines that we do have, what they're doing is they try to induce an immune response in people vaccinated where they'll make antibodies that will target a protein. I'm sorry, I don't have a pointer, but they coat the surface of those little sporozoids. So it's chock-a-block full of this protein, and the vaccine is made from that protein, and the idea is you'll get antibodies that will then recognize them and clear them from the body. So this could be a very, very powerful vaccine approach. Now, Now this vaccine, the R21 and the RTSS vaccines have been begun to be rolled out throughout sub-Saharan Africa, immunizing young children. This is the dashboard from the WHO showing the current status of the rollout. And you can see anywhere in green, that's where these vaccines have been introduced in these countries. So we're very excited to see this rapid rollout of these vaccines and hope that they will potentially have a very profound impact on malaria control. But we also need to be cautious because these vaccines are not 100% effective. So what that means at the liver stage is that if any of those parasites still slip through the cracks, if you like, and avoid the immune response, they will go through this massive replication, producing possibly thousands of parasites that emerge from the liver and go on and infect the red blood stage, the red blood cells. So there is this push and pull between the efficacy of the vaccine, but also if they don't protect, then they can still go through these stages. Now, Linnie also mentioned that another challenge with the liver stage is we have no way of diagnosing it, and we have no way of giving anyone prophylaxis, particularly for falciparum, for this particular stage. Let's move on to the blood stage. So this is the stage that causes the clinical disease. Linnie talked about some of the interventions in terms of drugs that will support patient management. And this is really important that it's coupled to good accessible diagnostics, because we don't want to just treat everyone as presuming that they have malaria. They could have something else. They could have a viral infection, a bacterial infection. And if we give antimalarials indiscriminately, we're promoting drug resistance. So we now have rapid diagnostic tests, which were a real game changer as an intervention because they could be used in low resource settings. These are like a pregnancy test or a COVID test, or you take a drop of blood and you can detect a protein that's produced by the parasite, and you can pick that up in the blood. So that's been a really, really key intervention, particularly in low resource settings. And we can also look at public health interventions for the blood stage. And now we're talking about chemoprevention. And what that means is giving antimalarials to targeted populations to try to control epidemics or to try to protect the most vulnerable populations. And this is not a new idea. This is a photo from Italy in the 1930s where people are being given antimalarials to protect themselves from the disease. And now this is an approach that's widely used, particularly in sub-Saharan Africa. And it can be focused on certain populations, for example, pregnant women or infants, school-aged children. It's also being used in really interesting ways based on the seasonality of malaria. So in many African countries, there's clearly a dry season with no malaria transmission or low transmission. And then once the rains come and the mosquitoes start to breed, you get this window when people are becoming highly exposed to malaria. So an approach called seasonal malaria chemoprevention will give people antimalarials right at the beginning of that rainy season and protect them throughout that really dangerous period of the year. So those are all very effective strategies at the community level. But they also face threats. And the number one threat is the emergence of drug-resistant parasites. And I'll also briefly mention how these rapid diagnostic tests may be failing in many parts of the world. So let's look at drug resistance. Now we're talking about parasites resistant to our frontline antimalarial. This is artemisinin resistance. So what you're seeing in this map is all those countries in red are where they've reported that parasites are developing resistance to these drugs. Now, it doesn't mean these drugs don't work at all. It just means they don't work as effectively as they did 10 years ago, for example. So we really need to be vigilant and find new drugs and alternative ways to try to protect people in terms of patient management, but also for using these drugs for public health interventions. Now, I just want to mention about this issue with the diagnostics because I think this, Well, it certainly caught me by surprise that this happened. So these are these tests that detect a protein that's expressed by the parasite. And it was first observed, I believe it was in Peru, that people had a test that said they did not have malaria, but when you looked under the microscope, you could see that the parasites were there. And so through some genetic sleuthing, they discovered that some of these parasites had actually removed the gene that encodes the protein that was detected by these diagnostics. So it was an adaptation of this parasite. Again, this parasite wants to survive as a species. So it evolved to no longer make this protein that the diagnostic test would pick up. So this allows the parasites effectively to fly under the radar. They're not detected by these diagnostics, and therefore that patient won't be treated, so they can live happily in their host. So this is very concerning, and we're seeing these genetic changes in the parasites in many countries, now in Ethiopia as well, and this really threatens the efficacy of these diagnostic tests. Okay, the last stage where we can intervene is at the mosquito stage, and actually we have a number of interventions for this stage. We can try to prevent those gametocytes, those sexual forms, from actually reproducing in the mosquito, and there are vaccines in development that are trying to block that transmission. We can try to just get rid of mosquitoes altogether, and that was the strategy that was done in the 1950s, which was very successful in eradicating malaria from Europe, or at least eliminating malaria from Europe. And the idea there is just get rid of those standing pools of water that the mosquitoes like to breed in. We can also add chemicals to those waters that will kill the larvae from the mosquito. And there's even some exciting work being done on making genetically modified mosquitoes that will be sterile to get rid of that mosquito population altogether. But there's also some interventions that will address that top part, which is let's prevent people from getting bitten by mosquitoes. We'll leave mosquitoes, allow them to live, but we don't want them to bite anyone. And some of those strategies include what's called indoor residual spraying, and that really leverages the biology of this mosquito, which likes to feed indoors late at night. And when this mosquito comes in, she likes to rest on the walls. So you spray the walls with an insecticide. So when the mosquito goes to rest, she's exposed to toxic chemicals. And the other similar approach is to have bed nets. And bed nets is a very important intervention against malaria. So the idea here is that people will sleep under a net, and that net is now a physical barrier to prevent someone from being bitten, but those nets are also treated with insecticides. So again, the mosquito comes in, she lands on the net, she can't get through, but she's also exposed to these toxic chemicals. And this was a recent systematic review that reported the tremendous impact on bed nets on child health. So reducing child mortality by 17% and severe malaria episodes by 44%. But as I said before, the mosquito also wants to survive. And so there is this push and pull. And what we're seeing is widespread resistance by the mosquitoes to these insecticides that are used for spraying, that are used for bed nets. We're also seeing some very concerning behavior change with mosquitoes. And of course, layered on top of that is we have the effects of climate change. So this map here is showing the reported mosquito resistant to insecticide. So everywhere where there's a red dot, that means there's confirmed resistant mosquitoes. And where there's an orange dot, that means a very high probability that those mosquitoes are resistant. So you can see that globally, there's widespread resistance in the mosquito population and a very high density of reported resistance in sub-Saharan Africa. Now, we're also seeing a new mosquito in town, if you like, particularly in sub-Saharan Africa. And this is a mosquito called Anopheles stevensi. This is a mosquito that was native to South Asia, but is now emerging in Africa, particularly in the Horn of Africa, but also reported in West Africa. So why are we worried about this mosquito? Well, the behavior of this mosquito is a little bit different from the traditional mosquitoes that would transmit malaria in Africa. And the main difference is that this mosquito likes to bite earlier in the evening instead of late at night, and it likes to bite outdoors instead of indoors. So what that means is that children playing soccer in the community after dinner are now being exposed to malaria much earlier on. Also, it means that those bed nets that protect people from being bitten late at night may no longer be as effective. And just to give you an example of the impact of this new mosquito, let's look at the case numbers from Djibouti, which is one of those countries in the Horn of Africa. So these are the number of cases in Djibouti from 2010 to 2020. And you can see in 2012, there were very few cases of malaria. But then Anopheles stevensii came into play. And then look what happens. You see this dramatic increase in the malaria incidence because now these bed nets are no longer effective, and we're seeing exposure to this mosquito. Now, the last factor, which is kind of permeating all of this, is the effect of climate. So these mosquitoes like to live in a certain temperature range, a certain range for rainfall. And as we go through climate change, we're going to see that sweet spot for the mosquito is going to change. That might benefit some countries where it actually becomes too hot to support transmission, but the reality is that it's going to mean many countries that haven't seen malaria for a very long time are now going to see malaria come back because the mosquitoes will be there. It also impacts some of those interventions that are based on this seasonality of malaria. If we lose that seasonality of the dry and rainy season, we can't use those interventions anymore to protect people in that way. And also when we have extreme weather events, this causes flooding. This can create new breeding grounds for the mosquito. And many of you will probably recall that just a few years ago, there were outbreaks of malaria with local transmission in the southern United States, in Florida and in Texas. So I think this is a really important reminder that these mosquitoes are still here. And as things change, we need to be very vigilant about this resurgence of malaria. We need to keep the foot on the brake, if you like. And with that, I'm going to pass it back to Linnie to take us through some of those broader challenges in malaria control.
Thank you. So, broader challenges. Here we are now. What can we do? What are we facing? Well, malaria is a disease of poverty, and it perpetuates poverty. Places where we have weak health systems, they fail to support diagnosis and treatment. People die. Limited resources are available for infrastructure that would limit transmission, electricity, window screens. Many of you are here to supply those critical bed nets. Remote and indigenous communities and those experiencing conflict, wars, people immigrating, are most vulnerable. How do we reach them? What can we do? And malaria, make no mistake, perpetuates that poverty. You can't go to school. You're ill today. You can't get medicine. How can you aspire to do more when you're not there to learn? Lapses in employment can lead to families without security that they need in order to progress, in order for whole countries to move on, as many of you know. Those healthcare resources have a national economic impact if a lot of the money that you would need goes to combating a disease that's ever recurring. And the mental health consequences, which many of us are aware of, in terms of the uncertainties of how to move forward. So, let's take this all, and I know you're here because you're already down to this call for action. As we've already gone through several graphs, we were winning, we thought, maybe not as well as Paul Russell thought, but we were winning. But now this is where we are in the world, at least of a couple of years ago. And I think you're going to hear today that maybe the data shows that that line is yet rising. As Pedro Alonso said in 2022, in his letter to his partners, he said, 24 countries have registered increases in their malaria mortality rate. The trends seen in sub-Saharan Africa are especially worrying. Malaria cases and deaths remain unacceptably high. And the hard truth is that we have approximately the same number of cases today as we did 20 years ago. Now, that was in 2022 from the head of the World Global Malaria Programs. We're now in 2025. But I think what he said then, that we were facing a malaria crisis, is ever true at this moment. So, how are we going to go forward? We're going to talk a lot about that today, about how attention to the disease is needed more now than ever, how we can't just do one thing, as Stephanie went over. There are many points of intervention, and we all need to figure out where are we going to try to make our mark together, and that they have to somehow be sustainable. Otherwise, we have this zigzag up and down, as has gone on for the millennia, and certainly since we've discovered the entire life cycle at the beginning of the 1900s approximately. What can we do? Well, community engagement. Engagement like we have today in this room. And I'm here in this room because of community engagement. Where's that Bryna and her lovely husband? We met at this place. As mentioned, I'm the immediate past president, and they were at a booth, and they said hi, and I said hi, and we talked, and they talked about malaria, and we ended up with them inviting me to come here. Like minds. People interested in making a difference in the world, doing what they could. And so I'm here now, happy to be members with all of you in this fantastic organization that is doing so much. I want to extend an invitation for you to come to our meeting, which will also be here in Canada, in Toronto in the fall. We are the largest international scientific organization of experts dedicated to reducing the worldwide burden of tropical infectious diseases. And come, join us. And I want to join more with you and all of the organizations you represent, and let's find others to bring together, because we can make a difference, each and every one of us, and each one we can bring into the fold. And it doesn't matter what any government is doing or any side is doing. Let's help save lives. Thank you. And I must give special thanks to the husband of my lovely colleague who made these beautiful slides. I have a few gray hairs, and these are absolutely the most beautiful slides I've ever used. Can we give 3 cheers?
This is an opportunity. We thought it would be kind of fun for people who had questions, but it's sort of hard when you're talking about science, and a whole lot of people are going to talk about these things later in the day. But I looked at some of your graphics about where the disease exists, and I wanted to know if you'd like to comment any further on the science behind that and how we promote scientific research in those areas that inform the local group, the local health personnel, how they're going to proceed. How do we do that?
Well, I think we have education on all levels, in all places. I'm trying to figure out what you told me about the camera.
What?
Are we on this end or that
Over
end?
that side.
Move down a little bit.
Yeah.
I always like to keep them happy.
Go ahead.
Yeah. I think that's critical, that you have education of the people on the ground at all levels. As a physician, I think the tendency to underestimate the importance of that, no matter what disease you're facing, is important. And so for people to understand what is the life cycle, that there may be stages when people don't know they're infected, that pregnant woman may think her pregnancy is fine, but it's not, that your little child that you think just have a cold may have a deadly disease, and what is the importance, as many of you do, in terms of bed nets and in terms of using the diagnostics when they might be wrong. So I think that never underestimate the power of people who are educated, and make sure that we do that.
Thank you. I'd like to invite anyone in the audience to ask a question. This is a time we have our scientific team. Thank you. Please state your name so that we know who you are and where you're from.
I'm Chris Roselle, and I work with village Rotarian women in Yumbe, Uganda, which is on the border of South Sudan. And in RDT community surveys, we found communities with as high as 92% of the entire community with malaria on the day of the survey. And my background is public health and ecology. So one of the things about, and correct me if I'm wrong, about parasites is they strive not to kill the host, and the genetics is to select for the ones that don't kill the host. So the most severe problems, I think, of malaria are the ones you mentioned of poverty. And we found that our communities spend as much as 50% of their income, and I know the data isn't, our economic data isn't great, but they spend as much as 50% of their income treating malaria, not counting days lost, not counting, et cetera. And so I'm wondering what to do with immunizations and bed nets, and if I shouldn't mention it, but we know also that malaria is not just, the mosquitoes don't propagate in just any water, they propagate in swampy, stagnant water. So what did we do?
Thank you.
Thank you.
Is this on? Yeah, okay. Well, I mean, when we talk about malaria control, we talk about our toolkit. And I think we need to be mindful that we have many interventions, but it's not just one magic bullet. So we need to think about how we invest in the toolkit, but also the delivery of that toolkit that has to be customized for the needs of that community, whether it's combining bed nets with the new vaccine, with drug treatment, diagnostics. Unfortunately, as much as we would love to have just one tool and say, let's invest in that, I think it needs to be thought of as this toolkit that will be appropriate for that community. And in an area like South Sudan with conflict and so many other challenges for public health, it needs to be properly integrated.
Dr. Gollightly, could you comment? This poverty is everywhere.
Yes, poverty is everywhere, and I think that what Stephanie just said is critical. To try to find where is the spot and how is the way that you can work together in a community, with a community, to try to break that cycle of poverty, and that on the one hand, you need to detect and treat, and on the other hand, if you could prevent that from ever occurring, you would save those funds. So what is going to be the most impactful things that you can do working together to try to stop it at some point, and then build on the money saved to then reinvest in continuing that? Again, it has to be something sustained. And that takes a long-term committed effort of dedicated, doesn't have to be many, but people that are really going to work towards doing that. And then I think, though, that that's in how you get lifted out of poverty. If you're not spending 50% of your income every year on malaria, you can put that towards other things.
Other questions? Could we have a microphone? You got a microphone in the back. Thank you.
Yes, hello everybody, and I want to greet you here from Germany. My name is Martina Möllers, I'm an epidemiologist. I'm working more than 20 years in sub-Saharan Africa so far, and I did my research with the London School for Hygiene and Tropical Medicine in regards to malaria prevention during pregnancy. And I'm working in different settings, in West African setting as well, in East African settings as well in South Africa. And my point is, regards of the resistance, what we have seen all over nowadays by malaria drugs. And we have a German pharmacologist with whom we have been closely working together, and I'm representing here as well as Area Coordinator Africa, our biggest and oldest RMCH Rotary Action Group for Sexual Reproductive Maternal Child Health as well. The case is, what I want to emphasize, we should really bring it into consideration. There is a plant calling Artemisia annua, and this is regards of the, I would say, when we are going from the drug production point of view, as well we can including as well some herbal stuff to make an impact, and more than less, we have seen it, we did some research in the Congo side, that we can really, even vulnerable communities, save in certain ways, that they don't really have the challenge or the burden for sure of malaria regards of all these resistance areas. We can combine it. And I think we have to look out of the box and to make sure in a holistically approach we can change something, because the drug stuff with the resistance is similar like with antibiotics, which we are seeing over these last years. I think, is this discussed, or is this still even an option for you? This is my question here. Thank you so much.
I'm not sure if I fully
So-
caught the question.
So as I understand the question is what are the roles of plants that could be used in the setting of prevention and affecting, I presume, primarily the insect vector and as a vector control approach?
Oh, is that what you were referring to, or for anti-parasite drugs? Yes, I would say yes. Anti-parasite. Yeah.
Okay. Okay.
Well, our classic malarias all come actually originally from plants, if you will. Quinine, cinchona bark, Peruvian bark, all of our lines come from there. Artesunate is an ancient Chinese herbal medication, if you will, original derivative. So I think the key is to investigate new drugs or purported drugs and see, do they work? What are the active components? And then incorporate that into our pharmacy that we use for malaria. So I think that we have one of the issues with resistance, which of course is a problem for all infectious agents, that eventually you end up with organisms that are resistant to whatever we're using. We are actively looking, and should be actively looking, for new root drugs, if you will, or root agents. So I don't think there's any problem with that. I think it's trying to find out what is the active component, and then understanding what are the side effects, importantly, and how they might affect the human host, because there's always good things and bad things about any drugs.
Yeah, and
Hang on for Dr. Jago's presentation in Malaria 301. We'll certainly be talking about that extensively a little later. Other questions? We have a...
Sorry. Thank you. Hi, my name is Julie, and I'm from Minnesota, an epidemiologist who's working in Sierra Leone. And my question is for Stephanie. I'm wondering if you can give us an update on malaria vaccine that may be available in pregnant women. And now for about over a year, we've had widespread rollout of vaccine in children. We're working in an area where there's no adverse event reporting. Can you give us an update on that?
I can try. As far as I'm aware, the R21 and RTSS vaccines are not being given to pregnant women, although I'll defer to Sir Adrian Hill if there's any movement in trying to include pregnant women in the rollout. There have been some recent clinical trials with the other vaccine strategy, which is with this whole parasite, whole sporozoite vaccine, giving the vaccine to women preconception, and then looking at once they become pregnant, was there any positive benefit from having that vaccination? And it did suggest that there could be some protection. Now, that's not an approved vaccine yet. It's at later stage clinical trials. There have been some other experimental vaccines for malaria and pregnancy that went through phase 1 trials, but they did not elicit very broad protection against the variety of malaria parasites that are present in different areas. So I think that's a bit of going back to the drawing board and trying to design more complex vaccines to target pregnant women. So it's a little bit of a wait, I think.
Thank you. Another question?
Yes, good morning, everyone, and thank you for the presentations. My name is Yanow, and I head the malaria program in Nigeria. Mine would not be a question, but like a comment. I know that some kind of reference have been made on education, and I just wanted to also add my voice. Talking about education and research, there's an aspect that from us, from the malaria community and having, this wonderful organization, in our midst, it's important we begin to look at the social behavioral change. I know I had already started talking about it with a couple of colleagues, and even with this year's World Malaria commemoration, which had one of the themes, slogans as reimagine. Reimagine. We really need to begin to see malaria as really what it is, a dangerous disease. For most of us, in the clinics, we see even the clients, our patients, refer malaria as my malaria. Okay, so this is, and I think that this is a good strategy, a good way for us to begin to look at how to, get on board both the healthcare workers and even the populace to understand, because the two groups would have to understand the fact that malaria is dangerous. For us, the healthcare professionals, most times we look for the cause of fever and we really don't see it, and we just, diagnose malaria just for that. We know those are the things that are going on. And then coming from the other side, they are in a hurry to embrace malaria. That shouldn't be. I think that if there's a research, that will be going through that path to begin to reorientate, maybe get, all of that, perspectives from the both groups, and then we'll begin to reorientate seriously. Then people will start taking it serious. I think that, for example, people cannot say I have my HIV status or something, There's always that stigma, but for malaria, it's not the same. So I wanted to just flag it here so that maybe that will be part of what we can have topical discussions on this beautiful event that we've all gathered for. Thank you very much.
Thank you so much. And as we are all aware, in the organizations that we represent, that nearly a quarter of the deaths in sub-Saharan Africa are in Nigeria. And we must have excellent research in that area. And you're asking about the social and behavioral change research that is required to understand that. We'll, in our session at the end of the day, Dr. Jimmy Opico is going to talk a little bit about how that part of the process is worked on through national malaria programs. And I think that that's a very important part. We need some science and we need some implementation. And we hope that Rotary can play a significant role in promoting and advocating for the kind of behavioral change you're talking about. Thank you. Another question. Two more questions.
My name is Niji Raji from Nigeria. I'm currently team lead tackling malaria and malnutrition 3 states in the southwest Nigeria. I just want to provide an information. In your presentation, you did not indicate that Nigeria has started using the R21 vaccine. And right now, 2 states are doing a pilot program, Kebbi State and Akwa Ibom State. I want you to just note that. Thank you.
Okay.
I'll have to make sure it's updated on the WHO map.
Another question.
My name is Derek Pasternak. I am from Seattle, Washington, and I work with Malaria Partners International to look at the world literature on the subject of malaria. A previous comment was that it's an evolutionary failure for a parasite to kill the host. The parasite wants the host to survive so it can propagate and move on to other hosts. In this connection, in endemic countries, literature shows that anywhere between 10 and 25% of school children are tested positive for malaria, even if they have no symptoms whatsoever. And with the failure of diagnostics right now, with the rapid diagnostic test being ineffective in some communities, there's a development of different RDTs. So my question is, do we know how or when WHO is going to move to to recommend the new diagnostic test, which exists already, that circumvents the problem that we have?
Dr. Gollightly, do you know?
I don't know. Is there someone here who might know from the WHO?
We may hear more about that as we proceed.
I mean, we do have other RDTs already available that target different proteins from the ones that are now starting to fail. And those are we talked about the HRP proteins, but there's also LDH, there's a few others. So those have already been implemented, but often those are not as good for distinguishing which species of malaria that person's infected with. So we need to look at, I think they're calling them next generation RDTs that will be more specific, but also address this issue about the diagnostic failure.
Okay.
Bill, we actually have time for probably one or two more questions.
One more question.
Thank you very much. More a comment than a question. Ross Smith from Australia. My district works with Griffith University on a freeze-dried vaccine emerging slowly. When I did one of my first BedNet programs in East Timor some years ago, we trained, as part of the day, we would train all the mothers in the villages how to use the beds and the nets, because they have to be tucked in and not sleep against them and so on. But I said to these women at lunchtime, are you aware why we have the nets coloured blue? And they said, oh, yes, yes. They said, we know that if we see a blue net out over a fruit tree, we see a blue net in a creek, we tell the men to get that damn bed back on our children's beds. She looked at me and she said, in our country, the women do everything that matters and the men do everything else. Ladies, thanks for a great start to the meeting today.
Thank you. That's a good way to end this.
Thank you. Thank you all. We're going to take a brief break now and refresh your coffee cup, please, and come on back at 8:45. We will begin our keynote presentation. Thank you.